Okay, good afternoon, everyone. So today, what we're going to do is we're going to first do the lecture, and then at some point, probably about half hour before the class ends or somewhere around there, the TAs will bring back the exams. And I just wanted to remind you, in case people like grab their exam or whatever, and or just leave without looking at it, whatever. What you want to do is you want to get your exam. Look at it, you need to turn it back in so we have all the exams. But if you want a regrading of the exam, this is all in your course information, and I'll probably read this again later when we give this back to you again. But only the professors then regrade the exam. Okay, so if the TAs go, oh, maybe you get another point, a couple points there, whatever. No, you got to turn it into the professors. And since this was written by Dr. Polinkanov, you can give it to me, and I'll give it to him, or you can give it to one of the TAs. So what you want to do is, when you see your exam, you want to write on the front of it. See, requests for regrading must be given in writing on the front page of the exam. So what needs to be regraded and why? So not just like, oh, look at question 2 again, but what was wrong with the grading of question 2. Okay, so if there's not enough space, you can staple another page to the front, and I have a few little pages here we can attach if you need a piece of paper. And for anyone listening to this, the recording later, if you were not here today or could not stay and get your exam, you can ask one of the TAs to bring your exam to one of their office hours, and you can look at it then. And again, if you want it regraded, you need to write on there to give it to the professor for regrading. Okay, and you have, so assuming they get here in time and everything, and we return them today, you have 2 weeks from today to request regrading. Okay, so again, this is all written out in the course information. What the policy is and how to do that. And you can, again, today when you see the exams, you can write that right on the front if you want to regrade, and you can give it to me or the TAs, and we can turn it into Dr. Polinkanov for you. Okay, so let's go on to here we go. Here's the lecture. Okay, so what we want to do is we started this whole big concept of glycolysis last time, and we're going to go through the rest of it. We started going through the pathway. Again, any structures and any information from the books, from the slides, from the homework, from the practice questions, whatever, is sort of fair game for the exam. But I will tell you that some of the things I almost always ask are, draw this step in glycolysis or draw that step in citric acid cycle or something like that. So I know some of you want a little extra time to really get to know some of these pathways. So I say up front right now, even though our exam's not for a couple more weeks after spring break, that you really want to know glycolysis, you really want to know citric acid cycle, but you definitely want to know some of these other feeder pathways and other phase pyruvate and all that as well. But if you want to practice drawing the pathways, you might want to start with glycolysis and citric acid cycle. Okay, so last time we talked about some of the importance of glycolysis. Okay, for some organisms, it's like the only way that they can get chemical energy of ATP for doing all these different things like movement and making ion gradients and things like that. And historically, glycolysis is very, very important, but it's also it's just this really important central pathway in metabolism. A lot of things feed into it. It feeds into a lot of different things, and it's a really important pathway in the cell, and also for learning about pathways and regulation and enzyme mechanisms and all different concepts like that. So that's why we go through this pathway, and also later the citric acid cycle in some detail. Okay, so we talked about the idea that we have 10 steps And to help us think about what's going on in these steps, they're often described as two different phases, the first 5 steps and the next 5 steps. So we have the preparatory phase, and that's basically the idea that we need to get all these reactions going. So we're actually going to invest some energy. We're going to invest some ATP, phosphorylate glucose, and then later phosphorylate glucose 6-phosphate, and get everything going. Okay, and it's in the second half where we actually start making some ATP. And we also make NADH too, which in some organisms like us can be used to make more ATP. Okay, so let's see, where did I stop last time? I was somewhere in the aldolase. I went to aldolase, right? We were talking about the splitting the and counting where the different atoms go. Okay, so just to remind you, we have, hexokinase. Hexose is a 6-carbon sugar kinase. Often transfers a phosphoryl group to something, and often from ATP. Okay, so hexokinase, great, glucose to glucose 6-phosphate. Okay, investing some ATP. Then we're going to do an isomerization, glucose 6-phosphate, fructose 6-phosphate. We're not adding on or removing any functional groups. We're moving electrons around. Same number of carbons, same number of oxygens, same number of phosphate atoms and phosphoryl groups and all that. Okay, this is an aldose to a ketose isomerization. Okay, and then we're going to invest some more ATP. And as I mentioned last time, this is the step where once we do this, once we take that fructose 6-phosphate and make fructose 1,6-bisphosphate, this product, this fructose 1,6-bisphosphate, is going to go through glycolysis. Before that, the glucose can be used for a lot of things. The glucose 6-phosphate can be used for a lot of things, and the fructose 6-phosphate can be used for a lot of things. Okay? But once you get past this step, you're going to go on through glycolysis. You're going to break down this molecule and get some energy out of it. Okay? So it's this step where if we want to turn up or down the activity of this pathway, this is an important step for regulation. This is what we say is the committed step. Once you pass this step, you continue on through glycolysis. You are committed to actually breaking down this molecule. Before this, you can store the molecule, you can build things with the molecules. After this, you're committed to breaking it down. So, the cell can sense. If I already have a lot of ATP, maybe I don't want to break down my glucose, maybe I don't want to break down my fructose 1,6-bisphosphate, maybe I've got a lot of ATP around, maybe I can build stuff, maybe I can make proteins, maybe I can make more cell membranes and stuff, maybe the cell can divide, right? Grow and divide. So this protein, this enzyme can actually sense when there's a lot of ATP around. The ATP will actually bind to this protein and slow down the activity. Okay, so ATP is both a substrate and also there's a way of binding to the protein to regulate its activity. Okay, so ATP is one molecule that proteins in this pathway and other pathways can bind to and sense that the cell already has a lot of energy. So we'll talk about regulation, about glycolysis, citric acid cycle, and other enzymes as we continue through some of these different enzymes and pathways. Okay, so we've got a committed step. We're going to go on. Now we're going to take aldolase. Now we're going to break down that fructose 1,6-bisphosphate, 2, 3 carbon molecules. Each one gets a phosphoryl group. Glyceraldehyde 3-phosphate will be used as a substrate in later steps, okay? But we have this other molecule, dihydroxyacetone phosphate. Well, fortunately, we have another enzyme that will take that dihydroxyacetone phosphate and make another glyceraldehyde 3-phosphate. If you look at these again, this is actually a ketose to aldose transformation, okay? No functional groups lost or gained. Same number of carbons, same number of oxygens, same number of phosphate atoms. We still have that phosphoryl group. And this is triose phosphate isomerase. In fact, the enzyme mechanism is very similar to the mechanism of phosphohexose isomerase that we talked about earlier. Okay, so only glycerolide phosphate is going to be the substrate for the next step. So we have to take that other three carbon molecule and make a second copy of glycerolide 3-phosphate. That also means that as we go through the second half of glycolysis, where we have these 5 more steps, we're going to have 2 of everything. We're going to have 2 glycerolide 3-phosphates going through the next half. Okay? Okay, and then this is, I think I finished up with this sort of concepts here last time. So this is our open chain form of fructose 1,6-bisphosphate. Again, you can draw this either as a cyclic form or the open chain form. You get both of them in solution. And so this is pointing out how we number the carbons, 1, 2, 3, 4, 5, 6. And if we chop it in half with aldolase, we get our dihydroxyacetone phosphate, a glycerolide 3-phosphate, and these are the numbers where it came from. Okay, so 1, 2, 3, 4, 5, 6. And if you kind of flip this molecule over so you see the matching phosphoryl group and make the other copy of glycerolide 3-phosphate, you get 2 glycerolide 3-phosphates. So in the molecule glycerolide 3-phosphate, yes, they're numbered 1, 2, 3 like this, the carbons, the 3 carbons. But these are the numbers over here on the left of where they came from in fructose 1,6-bisphosphate. Okay, so there are, don't remember what I put in the homework this time, but certainly if it's not in the homework, it's in the other practice questions. Following an atom from, say, glucose, or maybe from fructose 1,6-bisphosphate, if I say, okay, most carbons on our planet are carbon-12, but occasionally there's some carbon-14, okay? And you can buy molecules like this, glucose or fructose 1,6-bisphosphate, whatever, that were specifically made so that a carbon in a particular position is carbon-14 and the rest are carbon-12. And then you can do various things to follow what happens to that carbon, where it ends up, as you continue through a biochemical pathway. So you can say, oh, if I have carbon-14 at position 2, and the rest are carbon-12, then we chop this in half with aldolase. We get this one right here will be labeled with carbon-14 and none of the rest. And when we take this dihydroxyalone phosphate and make glycerolide phosphate, we get this molecule, and that means half of the molecules will have carbon-14 at position 2 and glycerolide 3-phosphate right there, right in the middle there. Okay, so you can do that with any of these atoms, figure out where they'll end up glycerolide 3-phosphate. You can continue through glycolysis then. You can say, okay, it's there in glycerolide 3-phosphate, and then you can see where it is the next molecule, and the next, and the next. And you can even follow it on through things past glycolysis too. The last one will be pyruvate, and then what happens to pyruvate? Well, it depends what cell type you're in and whether or not you have oxygen and other things going on. There are many different uses of pyruvate. But you can say, oh, if I went through glycolysis and then I sent my pyruvate through such and such pathway, where does it end up in a certain molecule? You can label one and follow it all the way through. Okay, so this is the chart to help you understand that, and this question of where does a particular atom end up as we continue on through the pathway. And so you can start here, or you can start all the way back with glucose, and follow, carbon 1 in glucose is going to be carbon 1 in fructose 1,6-bisphosphate, etc. Okay, so anyway, so that's how you do those questions if you've looked at some of the things that I've assigned. Okay, yeah.
Any one of the carbons can be assigned to be carbon-14 or carbon-12, whichever.
Right. Right, yeah. So assume that I can go and buy whatever I want from the store, okay? My carbon-14 can be in any of the carbons. And I could even tell you we're going to do it with oxygen. So we'll use oxygen 18 instead of oxygen 16. So you could do that with other atoms as well, in addition to carbon. But carbon's one that we kind of use as our example here. But yes, oxygen, I can tell you, we're going to use an isotope of oxygen. That's not your normal oxygen 16. So that can lead to some fun and interesting questions on homework and stuff too. Okay, so now we've got 2 glycerolide 3-phosphates. We're going to go on to the next step. Okay, this is a very interesting. Here we have glycerolide 3-phosphate. We're going to make 1,3-bisphosphoglycerate. We're adding on another phosphoryl group. This is not coming from ATP, okay? This is not coming from ATP. This is being added on from inorganic phosphate, okay? We're actually going to help out this reaction with NAD. We're going to have NAD reduced to NADH, okay? And when you have reactions that, when you have a redox reaction, and often, if you have an NAD going to NADH or the other way around, or FAD going to FADH2, one of the clues that you might need something like that is if it's called a dehydrogenase. Okay, so glyceraldehyde 3-phosphate dehydrogenase. There's my redox cofactor, NAD to NADH. And this, very interestingly, also we're bringing inorganic phosphate. We're going to phosphorylate 1,3-bisphosphoglycerate. Okay, now this is great because this kind of molecule, look at that. Look what we can do with that. We can take bisphosphoglycerate and we can actually make ATP from ADP. Okay, so it's a very high energy compound. It has a high free energy of hydrolysis, even of that phosphoryl group, and even higher than ATP, so we can make ATP with it. Okay, so here's our 3-phosphoglycerate, and here's our ATP. Okay, note this enzyme is called phosphoglycerate kinase. Okay, now I kind of emphasized before the idea that a kinase takes a phosphoryl group, often from ATP, and puts it onto something. And you can have a protein kinase, you can have a sugar kinase, whatever, okay? But remember that sometimes enzymes are named actually for the reverse reaction of what we're looking at in a particular pathway. Maybe it was first found because it does the other opposite reaction, or maybe in other pathways it goes in the opposite way or something like that. So this is phosphoglycerate kinase, but you can see it's kind of, in glycolysis we're going from the top here to the bottom. Phosphoglycerate kinase from the name, kind of, you can see here's product 1, product 2, you kind of think of it as, oh, it sounds like it's going from the products to the reactants. So it is named for the opposite of what's happening in glycolysis. You'll just have to kind of remember that. Let's see, and then I jotted down some notes about that. So we have our 1,3-bisphosphoglycerate, and I think I emphasized before in one of the other molecules, when we say 1,3-bisphosphoglycerate, again from the name, so there's glycerate, okay, 3-carbon compound, acid there, and then you have a phosphoryl group, another phosphoryl group, okay, that's phospho, that one carbon, 3 carbon, and the bis means you have 2 phosphoryl groups and they're not attached to each other. So 1,3-bisphosphoglycerate, that's how you would draw that, okay. Okay, so we're down now to 3-phosphoglycerate. Got rid of one of the phosphoryl groups. And we're going to move that phosphoryl group to give us a molecule that's a little better. Eventually we're going to get a molecule that's a little better at giving up another phosphoryl group to make ATP again. So look what's happening here. 3-phosphoglycerate, 2-phosphoglycerate. Okay, so we have phosphoryl group on carbon 3, phosphoryl group on carbon 2. It's phosphoglycerate mutase. So let's see what's happening here. We have a phosphoryl group being moved. We're not adding any carbons, removing any carbons. We still have the same number of phosphoryl groups before and after, the same number of oxygens and everything. This is catalyzed by phosphoglycerate mutase. A mutase is a subset, it's a type of isomerase. So this is based on isomerization. the enzyme is called a mutase because it's actually has the effect of moving a whole functional group from one place to another. Okay, so it's basically kind of like the phosphoryl groups moving from the 3 position to the 2, even though that's not exactly, it doesn't just go up and move like that. This is a little more complicated mechanism, but that's basically the effect of between the substrate and product. Question over here.
So why does this reaction happen? Does it make the molecule more stable?
So what we're going to do now is we're going to take the 3-phosphoglycerate and we're going to do some things to it so that it can give up the other phosphoryl group to make more ATP. So it's going to benefit us a little farther down.
In that position, it is not able to give it up?
We need to move it and then move some electrons around, and then it will be we want to increase the free energy change of giving it up. So you can get it off of there now, but it's not going to be a high enough free energy change to make ATP. So that's a good question. Why not just take it off here? You know, so when we move things around, change some bonds and everything, it'll be a better donor to ADP to make ATP. Okay, so in this class, we're not going to go through a lot of detailed mechanisms, okay? But I wanted to point out one. This is a fairly straightforward one. There's only a couple steps. As an example of some of the things that are happening. In this class, we're mostly talking about here are the substrates, here are the products, it's the type of the enzyme. We're not really saying how anything happens, but I want to give you an example that's kind of an interesting one of some of the things that can happen in active sites. Okay, so this is an enzyme that uses what we call covalent catalysis, and it makes use of very specific amino acids in the active site, histidine, okay? And we talked about some of the amino acids getting phosphorylated. Mostly we talked about serine, threonine, and tyrosine, but histidine is one of the other ones that can also be phosphorylated. And what we have here is an enzyme active site that's kind of squished oval kind of shape or whatever. Here's a couple histidines that are part of the protein so you'd have the whole, polypeptide chain out here somewhere, and you have a couple histidines facing into the active site. And this is an enzyme that before it's active, it actually gets phosphorylated by another enzyme. Okay, so here we have a phosphoryl group on an enzyme histidine already. And then our substrate comes in, our 3-phosphoglycerate. And this other histidine can take off a proton. So to help help the 3-phosphoglycerate accept another phosphoryl group, you get 2,3-bisphosphoglycerate. Okay? And then that histidine up here is going to accept the phosphoryl group from the 3 position. And so what do you end up with? You end up with your product. Great. The other thing we end up with is our enzyme back at the starting point. Okay, so if you simply took a phosphoryl group and didn't give one back, you'd be using up the enzyme. It wouldn't be a true catalyst, right? So the way this works, you actually end up with the phosphoryl group on the enzyme, and the enzyme is ready to go catalyze another, the reaction again. Okay, so it's an example of how those amino acids in the active site can really help in very specific chemistry. They need to be positioned, of course, in just the right places, but it's a little more complex than what you might have pictured from when I just say here's the substrate and here's the product. But again, it's a relatively simple mechanism. I didn't want to go in too many really long, lengthy ones. Okay, so now we have, again, the enzyme's ready to go again. And now we have 2-phosphoglycerate, okay, still not quite ready to become the phosphoryl group donor. Okay, not quite ready, getting there. So now we're going to catalyze this next step by the enzyme enolase. So we're going to remove a hydroxyl group and a proton, give us a double bond there. Okay, so essentially we're removing H2O or water. And we end up with this phosphoenol pyruvate. And now this molecule is a better phosphoryl group donor. Okay, and now the phosphoenol pyruvate can give a phosphoryl group to ADP. And we end up with pyruvate and ATP. And notice again, the name of the enzyme kind of sounds like what you might call it if you're naming it for the reverse reaction. So pyruvate and ATP giving you phosphoenol pyruvate and ADP. Okay. This reaction has a fairly strong, high standard free energy to go forward. Again, we talked about this a little bit in the one of the previous lectures where we talked about why some molecules are better at giving up phosphoryl groups than others. And so this is one where you make that initial version of pyruvate, but then you have this tautomization, you have some movement of electrons and everything, and then you get a more stable version that's less likely to go back to substrate, and that helps drive the whole reaction to actually making ATP. Okay, so here it is again. I showed you this earlier when we were talking about chapter 13. Okay, so drive towards ATP formation. Okay, so that's all the 10 steps of glycolysis. Get to know them really well, get to know what's going on in there, not just kind of memorizing name, structure, but what's happening in the different steps. And in doing that, you can kind of think about what is the name of this, reaction, and it often kind of matches with what's going on. What is the purpose of the reaction, and this idea of following how a particular carbon goes through these different molecules in the pathway. Okay, but let's talk about overall what happened in glycolysis. So we had a glucose, okay, 6-carbon sugar. We ended up with 2 pyruvates. We also had some various other molecules come into play here. So we had some ADP, some ATP, some inorganic phosphate, okay. Overall, we used 2 ATPs, right, in 2 different steps up there in the first half of the pathway, but we made 4. So we ended up making 2, okay, pretty good. Not a lot of ATP, but it's fine for some organisms. This is it. This is their way of making ATP, and they survive, okay? We need more, but it's good for some organisms. Okay, and then oxidation reduction reactions. Remember we had that reaction where we made NADH, okay? That'll come into play later, too, when we talk about making more ATP in organisms like us. Okay, so overall, what do we have? We have glucose, we had some ATP invested, we had some NAD, 2 because, remember, the second half of the pathway we're doing everything twice. We made 4 ADP, 2 steps where we made them and going through twice, so that's 2 times 2 is 4. We used 2 inorganic phosphate, we got pyruvate, ADP, NADH, et cetera, et cetera. We can kind of cancel some things out. So here we made and used some ATP, so overall we used 2 ADP and made 2 ATP. And we can go back to some of those equations where we talked about, making free energy, or not making, I should say, reactions giving off free energy or using free energy and all that. So if we look at this overall reaction, we can split it into a couple reactions. So if we have glucose, and we have some NAD, we make pyruvate and NADH, the change in free energy, standard change in free energy of that is minus 146 kilojoules per mole. Pretty good, okay? And then we made some ATP. We made 2, so that's 2 times 30.5 kilojoules per mole, 61 kilojoules per mole. And remember, this is, since we flipped the direction of this, like usually if we're talking about ATP hydrolysis, we say it's negative 30.5, but since we flipped it the other way around, we flipped the sign, so this is positive. 2 times 30.5 kilojoules per mole, 61 kilojoules per mole. And then remember when we talked about in chapter 13, we can add these 2 free energies together. So we have minus 146 kilojoules per mole plus 61 kilojoules per mole minus 85 kilojoules per mole. Okay, so what happens? If we completely broken down glucose, we could get 2,840 kilojoules per mole, a lot of energy. Well, we got 85 kilojoules per mole, okay? So we got some energy from it, and again, some organisms are fine with that. There's still a lot more energy. If we kept breaking down these molecules, if we took that pyruvate and did more with it, we could get a lot more energy, okay? So that's where some of our later pathways come up. There's pyruvate, phase of pyruvate, citric acid cycle, things like that. So we're going to continue talking about breaking down things and getting more energy when we talk about the citric acid cycle. Okay, so there's still a lot more energy that could be obtained. I also want to talk about the idea that we don't eat just glucose, okay? We eat a lot of different types of food. So our bodies have evolved to make use of all those different things. Well, not all of them. Some of it doesn't do us any good, but some things doesn't do us any good. But a lot of different types of molecules can be broken down, and a lot of those molecules, those products then can be sent through glycolysis to get more ATP and to get more reduced NAD to NADH. Okay, so in this looks very confusing, but if you kind of look at the different parts individually, it starts to make sense. So up here, here's our glucose over here. Glucose, hexokinase, glucose 6-phosphate. Okay, first step in glycolysis. And if we continue through glycolysis, we go through some other molecules like fructose 6-phosphate, right? Fructose 1,6-bisphosphate and our good friend glycerol 3-phosphate. Okay, and then we can continue on through glycolysis. But these are some of the intermediates in glycolysis. Okay, so if we eat glucose, yes, we can send that through glycolysis. Okay, what about table sugar? Okay, table sugar is not glucose; it's sucrose. Okay, sucrose is basically a glucose, and that's covalently bound to a fructose. Okay. Well, the glucose, of course, we could just send through glycolysis, right? Hexokinase, blah, 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 all that, okay? But first we have to break it off with the sucrose, okay? So the sucrose is a sugar, ends in O's, right? Okay. The enzyme that breaks it down is sucrase, A-S-C at the end, sucrase, okay? So we chop glucose and fructose. The fructose can get into glycolysis in a couple different ways, depending on what cell type you're in for the most part, okay? So in some cases, great, hexokinase, we've seen that before. What does hexokinase do? Adds a phosphoryl group on carbon 6. Okay, wonderful. So we've got hexokinase, the glucose half of sucrose goes down to glucose 6-phosphate, the fructose goes to fructose 6-phosphate. Okay, so some of our tissues aren't that simple, they get a little more complicated. They take our fructose and they send it down this other pathway to make glycerol 3-phosphate. And it looks kind of confusing, but you know what? If you think about what happens in glycolysis and you think about the names of those enzymes and what are the chemical transformations that each one does, you could probably fill in blanks of what some of these names of enzymes are and what some of the products are. So first of all, if we take fructose, we're going to do something called fructokinase. So this is another kinase. It's going to use ATP, okay? And we're going to add a phosphoryl group. It's just that it ends up on the 1 carbon instead of the 6. So fructokinase is a kinase, but it's going to make fructose 1-phosphate, okay? Now we're going to take that 6-carbon sugar and we're going to chop it in half with an aldolase. This is a different aldolase than we were using before. Fructose 1,6-bisphosphate. I'm sorry, fructose 1-phosphate aldolase. Okay. And that's going to give us 2 3-carbon molecules like before. So we have a dihydroxyacetone phosphate. Great. We know what to do with that. Triose isomerase gives us glycerol 3-phosphate. The other molecule, right, this fructose 1-phosphate only had the 1 phosphoryl group. So we get glyceraldehyde, but we don't have that 3-phosphate. How are we going to add a phosphoryl group? We're going to use a triose kinase in ATP to give us glyceraldehyde 3-phosphate. Okay, so this is a little more complicated, but if you had to sort of fill in some of the blanks here, again, thinking of what the enzymes are doing, from the names, from what we know about what happens in glycolysis, I think you can kind of fill that in. Okay, so we have sucrose. We can eat sucrose, glucose, or fructose. Fructose also is just a sugar that comes from a lot of plants as well. We can also, trehalose is also made up of glucoses linked together. So we'll just chop that into individual glucoses with trehalase, again, ASE. Lactose is basically lactose and glucose linked together. So lactase chops that in half. You get glucose to glycolysis. Galactose has a slightly more complicated pathway here, but basically you're going to get glucose 1-phosphate. The glucose 1-phosphate, you want glucose 6-phosphate. We talked about an enzyme that does something like that already. You're going to have a mutase pick up a phosphoryl group, put it on the 6th position instead. And then over here we have mannose, which is very similar to glucose, has a lot of similarities. It's a 6-carbon compound, and let's phosphorylate it. What are we going to use? Let's use hexokinase again. Mannose 6-phosphate. We want to make fructose 6-phosphate. We have another isomerase. Again, this is a very similar mechanism to phosphohexose isomerase or triose phosphate isomerase. So we get fructose 6-phosphate. And then we'll talk a little bit more later about glycogen, which is a chain of glucose that we use for example, in our muscles and our liver to store a lot of glucose. And it's a big long chain, a big long branch chain, and we have an enzyme called phosphorylase that chops off one glucose at a time to give us glucose 1-phosphate. And then again, you can come down here with phosphoglucomutase and everything. We also have a starch in our diet from plants. It's a different sort of polymers linked together a little differently, so there are ways to break that down as well. Okay, so again, it looks really complicated. You might need to look at this a few times, but again, for example, some of these things that are sort of longer pieces, disaccharides, trisaccharides, whatever, the idea is you're going to chop them with something that has a similar name to the sugar. Okay? If you have a 6-carbon sugar like glucose, fructose, or mannose, a lot of times you can just phosphorylate with hexokinase, maybe do 1 or 2 other things to it to get it into glycolysis. And again, that galactose is a little trickier. I'm not going into detail about that one. And we'll talk about the glycogen a little more in one of the other lectures. Okay, so we can eat a number of other types of sugars, and what the cell does is it basically maybe makes a modification or a few modifications and just sends it down through a pathway that we already have, okay, down through glycolysis again, right into the central, one of the major central pathways of metabolism. Okay. So down at the other end of glycolysis, what happens? We get our pyruvate, ta-da, done. Well, something's going to happen to that pyruvate. It's not just going to sit there. We don't just have big piles of pyruvate everywhere, okay? We've got to do something with our pyruvate. And what happens to that pyruvate is going to depend on what kind of organism we are, okay? And also it can depend what cell type within that organism and the conditions that that cell is finding itself. And I'm sorry, I'm hearing some people talk. Let's hold our conversation till later. Okay, thank you. Okay, so if we're animals, that's us. Okay, that's us again, animals, and all our animal friends, okay, our cats and dogs and everything. Under anaerobic conditions, okay, and that doesn't mean we're holding our breath for a long time. That means we might have our cells have used up a lot of oxygen. Like if you're running a lot, your muscles might be a little short on oxygen temporarily until you catch your breath. Okay, it doesn't mean that we're standing here holding our breath for 10 minutes or whatever, okay? But when you run, some of your cells might temporarily not having optimal oxygen conditions, so it's anaerobic, okay, not so much oxygen around. Okay, so what happens? Think about it. You're running or you're swimming like many, many, many laps in the pool. What happens? Your muscles start getting a little tired, a little maybe achy, maybe sore, okay? So what's happening there? This molecule lactate is building up in the muscle. Okay, so our pyruvate is being converted to lactate, okay, using an enzyme called lactate dehydrogenase. Well, that doesn't sound too good. Why would you do that? Okay, why would we make lactate if it makes our muscles sore and achy? Okay, well, here's why. If you're running for the bus a long way, okay, or if you're swimming many, many laps, or if you're doing stuff where you can't get the oxygen to your muscles fast enough, you might not be able to use up the NADH that is made in glycolysis. NADH. NADH is used later in pathways that require oxygen to make more ATP. So if you're not getting all the way to those pathways, you don't have a lot of oxygen around, you're not taking your NADH and making new NAD. Okay? So then you might not have enough NAD around to really keep the glycolysis going, to make more ATP through glycolysis, to get your energy, to keep the running or keep the swimming or whatever. So we have glucose going to pyruvate, you make NADH. So a way to get some more of that NAD quickly, fast, while you're running, while you're swimming, whatever you're doing, is to use this lactate dehydrogenase. Pyruvate to lactate also means NADH to NAD, which means you have NAD, so glucose can be converted to pyruvate, and you can quickly be making some more ATP through glycolysis, okay? So this is a quick way of getting more NAD so glycolysis can continue even though you don't have some of those later pathways that require oxygen, regenerating the NAD and making more ATP that way. Okay, so this is quick, anaerobic in animals like us, okay? We're going to go from pyruvate to lactate. Now we don't necessarily want, remember that lactate's making us kind of sore and achy and everything? But what happens? You rest, you catch your breath. Hopefully that discomfort goes away. The lactate will actually move in your bloodstream to your liver and be used in gluconeogenesis to make glucose again. Okay, so if things go right, the lactate doesn't just hang around and make you sore forever. Okay, so this is what's happening when you're running for a long time. Or if you're old like me, if you're running for a short time, your muscles get sore. Okay, and this is why your muscles start getting sore after some exercise. You've got the lactate, you need to rest, and give it a few minutes, and your lactate will move out of your muscles to your liver and make glucose again. Okay, so do we have the exams? Do you have the exams? You have some of them. We're waiting for somebody else too? Okay. Okay, so we're going to wait a little bit. Some other people are coming with, there's a big stack, so we're going to wait for a couple of the other exams. Okay, now, other organisms have a lot of similarities to us. They have glycolysis. Most organisms have glycolysis. But they don't necessarily have all the same pathways we have, okay, like your friendly neighborhood yeast, okay? They don't swim laps in the pool, they don't run for the bus or anything like that. But they do have glycolysis, and under anaerobic conditions, again, when there's not a lot of oxygen around, they do something else with pyruvate, okay? They take the pyruvate, and in 2 steps, they make ethanol, okay? So you get pyruvate, acetaldehyde, oops, what happened? We lost a carbon. Okay, let me give you a clue. A lot of times if we're losing a single carbon off of a small molecule or a sugar or something like that, it goes into carbon dioxide. Okay? So pyruvate, Pyruvate decarboxylase, oh, it's right there in the name. Decarboxylase gives you carbon dioxide, gives you acetaldehyde, and then you have another dehydrogenase, alcohol dehydrogenase, to give you the ethanol, NADH to NAD again. Okay, so you can regenerate your NAD here. Okay, so this is nice. So yeast can also regenerate NAD, use up some of that pyruvate. Nice for the yeast. Nice for us too. Okay? We make use of this carbon dioxide. Why do we care that yeast are making carbon dioxide? What does it do for us? What is it good for us? Any idea when you add yeast to something to get carbon dioxide? Carbonation. So certain types of drinks, like champagne, you've got bubbles. This is what your bubbles are from. Pyruvate decarboxylase, carbon dioxide. Okay, there's something else. Think about when you're having a sandwich and you have fluffy bread, okay? Leavened bread. Causes bread to rise, right. So, again, pyruvate, acetaldehyde, carbon dioxide. Okay. A lot of people also like this part over here, the ethanol. Okay, so this is yeast, anaerobic conditions, beer and wine. Okay, so so this is something that, again, yeast share some pathways with us, like glycolysis. They tend to be less multicellular than we are. They tend to be single cellular. Sometimes they grow in clumps or whatever, but they're not true multicellular organisms. But this is a pathway we don't have, but they can make for us. They can make for us ethanol, and they can make carbon dioxide, and we use that in making certain types of foods and beverages. Okay. Okay. Are the TAs ready now? What? We're still waiting for? I can't hear you. Oh, we're still waiting for 2 people. Okay. We'll continue on here. We still have a few more slides in this particular lecture. Okay. So, if you're listening carefully to what I said, and a few slides ago, I said we make lactate in our muscles. And then that feeling of the achiness from the lactate goes away, hopefully soon after we stop running or swimming or whatever. Some of the achiness will go away. And that lactate, because it goes to our liver, and we can make more glucose. Yay. Okay. So that's a process called gluconeogenesis. Taking compounds, small molecules like pyruvate, sending them up, vertically up here, to make glucose. Okay? Why do we want to do that? Well, we're animals, and plants might have a lot of uses for glucose. Okay, so we talked about we can break it down again. We can store it for later. We can make things out of it. So here would be like blood glucose, like our brain needs to keep thinking, okay? We need glucose so our brain can break it down for energy, and other tissues as well. Store it as glycogen. That's us and animals again. Plants will make starch and sucrose, and we'll all make things like disaccharides and glycoproteins, other monosaccharides for a lot of different purposes and everything. So all different organisms need some glucose. And it can be made from a number of different sources. So again, here's animals. We can make it from a lactate. We can make pyruvate again, go up through some of these different steps, and make glucose 6-phosphate or glucose. And different things can feed into this too, in addition to lactate. Okay, different steps along the way. We can also take triacylglycerols. In plants, in this course we don't really go through this, but plants, again, they can take this carbon from the oxygen and make small molecules, carbon-containing small molecules that can be used to make glucose and other sugars and starch and everything. Okay, so the idea here is that we want to be able to take small molecules and make bigger molecules like glucose and other carbohydrates. So it's gluconeogenesis, so making glucose. Okay, so how are we going to do that? Paste all the little molecules back together again. How are we going to do that? Well, one of the things that you'll find in our metabolic pathways is that we reuse a lot of enzymes and a lot of molecules for more than one thing. So we have glycolysis that we just talked about in some detail, 10 steps, glucose at the top, break it down, break it down, break it down, all the way to pyruvate. Gluconeogenesis is going to just use a bunch of these enzymes in the opposite direction, okay? Which is nice. Fewer to memorize, right? Okay, great, okay. So we have these different pathways. When we talked a little bit about the whole idea of metabolism and everything at the beginning, remember we said that how do you have different pathways doing kind of the opposite things? Well, we sometimes specialize them. So like I was saying, your lactate usually goes to your liver to be used to make more glucose. So you might have gluconeogenesis going from the bottom of this figure to the top, more in the liver than in some other tissues. Whereas taking glucose and breaking it down and getting the energy might be more often used in things like muscle and brain that need a lot of energy. Okay? So different locations, different cell types might specialize in a pathway going in one direction than another. Okay, so we're going to use some of these same enzymes in glycolysis and gluconeogenesis. Which ones? Well, some of them, like glucose 6-phosphate to fructose 6-phosphate, this is just an isomerization. We're just moving some electrons around, okay? Here are free energy changes here, or these are standard free energy changes. What we want to think about is like, what's the actual free energy change in the cell, okay? Well, what happens is this transformation of glucose 6-phosphate to fructose 6-phosphate can be close to zero. Free energy change close to zero means you can just go in either direction equally easily, okay? A lot of these other steps, okay, fructose 1,6-bisphosphate, dihydroxyphosphate, glutathione phosphate, that was our good friend aldolase, okay? Again, near zero, you can go in either direction in the cell, and a lot of these other enzymes as well. Okay, so a lot of these enzymes, under cellular conditions, can go in either direction, okay? There's a few that don't really do that very well, okay? This hexokinase reaction, we're taking glucose, we're taking ATP, we're going to transfer a phospho group from ATP to glucose. That's hard to do backwards, okay? That's hard to do with the standard free energy change. It's even harder with the actual conditions in the cell, okay? Getting that phosphorus group off of the glucose back onto an ADP to make ATP, not really going to happen too often, okay? How about this fructose 6-phosphate plus ATP, fructose 1,6-bisphosphate and ATP? Again, going backwards, not going to happen too well, okay? And then this very last one, phosphoenolpyruvate plus ADP to make pyruvate plus ATP, nope, okay? It's going to be pulled in one direction. Okay, it's going to go in one direction, it's not going to want to go backwards. So, of our 10 steps in glycolysis, 7 steps can go backwards to help with gluconeogenesis. Okay, great. We just need to think about these other 3 steps. How are we going to do the opposite? Okay, and what we have, what we call the 3 bypasses. Okay, so 2 of these bypasses, we took a sugar, we took either glucose and ATP, made glucose 6-phosphate, or fructose 6-phosphate and ATP made fructose 1,6-bisphosphate. Okay? what are we doing in each of these if we're going to go backwards? We're going to take glucose 6-phosphate and make glucose. Or we're going to take fructose 1,6-bisphosphate and make fructose 6-phosphate. What is the general idea of what's happening here? Glucose 6-phosphate to glucose. What is the change in that molecule? Okay, you're losing a phosphoryl group, right? You're losing a phosphoryl group. What kind of enzyme removes a phosphoryl group? A phosphatase. Okay, so we have glucose 6-phosphatase. So again, the name kind of goes with what it's doing, right? Glucose 6-phosphate, remove a phosphoryl group, phosphatase. And note that, as happens very often with phosphatases, that phosphoryl group is lost as inorganic phosphate. It's not being stuck onto an ADP to make ATP, right? This is just removal as inorganic phosphate. Same thing happens down here with fructose 1,6-bisphosphate to fructose 6-phosphate, and that's fructose 1,6-bisphosphatase 1. The phosphoryl group just ends up as inorganic phosphate. Okay? So we get, if we look at our sugar substrates in glycolysis, we go from glucose to glucose 6-phosphate. We're using ATP. To go the opposite way in gluconeogenesis, we do not get our ATP back. Okay? Same thing down here. If we go from fructose 6-phosphate to fructose 1,6-phosphate, we invest ATP, but in gluconeogenesis, we don't get that ATP back. Okay, that's important. Down here is a little more complicated. Go from phosphoenol pyruvate to pyruvate. Remember, we used pyruvate kinase. We actually made ATP. To go back the other way, it gets slightly more complicated. We're going to take pyruvate, make an intermediate called oxaloacetate, which we'll see if you want to know how to draw it. We'll see that when we get to citric acid cycle. It's also an intermediate in the citric acid cycle. So we go from pyruvate to 4-carbon compound called oxaloacetate to phosphoenol pyruvate. So that's a couple steps. We actually, to do this first one, we actually need to invest some ATP, and to make the second step, we're going to invest GTP. Okay? Again, with glycolysis down here, yeah, we made a little ATP, but to go from pyruvate to phosphoenol pyruvate, we're going to invest ATP and GTP. Okay? So, so note a few things here. The names of the enzymes, what's happening in the different steps and everything. But the pathway of glycolysis and the pathway of gluconeogenesis are not exactly just opposites. Sure, we're going to use 7 enzymes in both of them, in either one direction or the other. But these 3 steps in glycolysis are not just the opposite. And basically, in glycolysis, we invest some ATP, we get some ATP, but then if you take the pyruvate and go back up, you actually have to invest ATP and GTP, but you don't get those ATPs back up here, okay? So you don't want to be going through glycolysis, getting pyruvate, and immediately sending it back up through gluconeogenesis, because the little bit of energy you get from glycolysis, you just lose that in gluconeogenesis. So we don't want both pathways going on in the cell at the same time, just continuously, because the net effect would you'd be wasting energy, you'd be losing energy. So these are going to be regulated. Under some conditions, maybe some cell types, maybe some of the time, you would do glycolysis. Maybe other conditions or other cell types or whatever, you'd be doing gluconeogenesis, okay? So again, you don't want to just be breaking down your glucose to immediately be building it up again. If you do if you do glycolysis, you want to have a reason for doing it. If you do gluconeogenesis, you want to have a reason for doing that, okay? And, okay, so I think Okay, so this is one last pathway. It's kind of important pathway. We're not going to go through a lot of details on this, but I did want to mention what it is and what it does. Okay, this is the pentose phosphate pathway. This is another way that we can use glucose. So here's our glucose. It's been phosphorylated by hexokinase again. We can send it down through the pentose phosphate pathway to make some molecule 6-phosphogluconate, et cetera, and make 5 carbon sugars. Okay, glucose again, 6 carbons, remember? Give off 1 carbon as carbon dioxide, you get ribulose 5-phosphate, and then ribose 5-phosphate over here. Hopefully you've seen the ribose 5-phosphate many, many, many times at this point because you need it for things like DNA, RNA, nucleotides, some of those coenzymes. Remember NAD, nicotinic adenine dinucleotide. You have a ribose, 2 riboses in that, right? Okay, so if you need some 5 carbon sugars like ribose, you can send some glucose through the pentose phosphate pathway. Another thing over here, this little bit over here, notice that we have NADP being made to NADPH, another one over here. This is a pathway that's not using NAD, it's using NADP. We're making NADPH, making more NADPH. NADPH is used in a lot of our anabolic pathways. If we want to make fatty acids, if we want to build up things like that, we need a source of NADPH. So this is a pathway where we can send glucose if we need NADPH to make things like fatty acids, or ribose 5-phosphate to make nucleotides, coenzymes, DNA, things like that. Okay, so again, you can send your glucose through different pathways. You can turn on and off different pathways. And for this particular pathway, the pentose phosphate pathway, it's going to sense, do we already have enough NADPH? Okay, this is one of the key things this pathway makes. So it has a way of sensing, do we already have enough? If we already have enough NADPH, you're going to use your glucose up here. Maybe you'll send it down through glycolysis, get some energy. Maybe you'll send it somewhere for storage as glycogen or somewhere else. But this is another example of regulation and sensing. What does the cell need right now? Does it need NADPH? Does it need ATP? Does it need neither? We just store glucose, or maybe we have lots of energy, lots of everything, and the cell can grow and divide and make more cells. Okay, so this is an example of regulation and another use of glucose. Okay, so I guess we can start handing out some of the exams that Hepeng has. I don't know where the other 2 TAs are. Oh, okay, Madu's here. Do you have all of them? You have all of them now. Okay, we're ready. Okay, let's do this. And remember, look in the course information about any questions about regrading. Only the professors can regrade, so, oh my, let's see. So please write down any regrade requests. Oh, okay, here we go. Regrade requests, look here. This is how to do it. If you want a regrade request, you need to actually write it on the exam and turn it in for a regrade request. You have to write down which question to be regraded and why. And only the professors can give additional points due to regrading. Okay, see you Thursday then.
Professor,
And you'll need to turn your exam back in to the TAs when you're done. Just separate it out if you need anything regraded. Okay? Thank you.